Hello students, now we will start with new topic, that is genetic code. Genetic code, it is defined as, it is a set of rules that is used by living organisms to translate the genetic material that is encoded in the genetic means nucleic acids or the genetic material into amino acids. We can write that one in the form of a definition. Genetic code is a set of rules used by living organisms or living cells to translate the information encoded in nucleic acids or genetic material into amino acids. First, George Gamow has given the idea of combinations of 4 different nitrogen bases into different patterns or different combinations to give out amino acids. George Gamow has proposed idea of 4 nitrogen bases arranged in different combinations to give 64 codons. And these 64 codons are nothing but they are coding 20 different types of amino acids. This idea initially was given by George Gamow. What are those 4 nitrogen bases? 4 nitrogen bases are adenine, guanine, cytosine and uracil. It is not thymine because George Gamow has taken some of the nitrogen bases from RNA. He did not take from DNA. Because here, if you see in the definition, translate genetic information from genetic material. We know that mRNA is undergoing a process called as translation to give out proteins or amino acids. But here, genetic material in the sense, which genetic material it has to be? It should be RNA. And the RNA that behaves as genetic material or having high accuracy to behave as a genetic material is only, there are tRNA is also having some information, but out of all the types of RNA, mRNA is considered as the best genetic material. So, the nitrogen bases present in case of mRNA are adenine, guanine, cytosine and uracil, not the thymine. So, George Gamow has proposed that 4 nitrogen bases that show some combinations to code for 20 amino acids. This is the proposition given by George Gamow. So, what are those combinations? He also proposed that combinations should be of 3 nitrogen bases. So, he strictly argued that 3 number of nitrogen bases out of the 4 different types of nitrogen bases are framing into one combination and they are coding for different types of amino acids. So this was considered as a correct proposition because there are 4 nitrogen bases. If all of them are combined 3 in number, so if we multiply like this, we get 64 codons. And out of the 64, 61 codons or combinations are coding for 20 amino acids. What about the remaining 3? Remaining 3 are called as stop codons. Means during the translation process, the formed protein is stopped at one point where it has reached a combination of 3 stop codons. It can be either UAA, uracil adenine adenine, or uracil adenine guanine, or uracil guanine adenine. When the translating proton has reached the stop codons which are present on the mRNA, then the synthesis of protein was stopped. So, basing on all the information that we are studying during transcription and translation has given a proposition and conclusion that George Gamow's proposition was absolutely correct. So, next, another scientist, Hargovind Khorana has developed a chemical method in 1970 that helped in understanding of RNA synthesis. Hargobind Khurana has developed a chemical method in 1970 that helped in understanding RNA synthesis. So for this purpose, what he has done, so he developed some types of oligonucleotides. What are oligonucleotides? If some types of, we can say copolymers, we can say Homo polymers, but here in this genetic material topic, if we discuss what type of monomers has been combined to give out Homo polymers or copolymers, it can be nothing but only out of these 4 different types of nitrogen bases. So this person, a chemical method we are telling, in the sense he has mixed some types of, with help of, so understanding RNA synthesis with the help of Homo polymers. We can take examples for Homo polymers as uracil, uracil, uracil. Same type. They code for phenylalanine type of amino acid. So he kept on mixing this. So when he has taken only one type of nitrogen base out of the 4, he has seen the formation of this phenylalanine amino acid. He also has taken copolymers in a combination of uracil cytosine, uracil cytosine, uracil cytosine, uracil cytosine, uracil cytosine. Here if you see, we are taking only 2 different types of monomers. Here same type of monomers. So we have used a term called as Homo polymer. It is giving out the amino acid phenylalanine. Phenylalanine is an aromatic amino acid. Though he has used 2 number, means more than 1 number of monomer or more than 1 type of monomer or 2 different types of monomers only, then also he observed the combinations of the codons that are giving out amino acids like serine. Cytosine, uracil, cytosine, giving out another type of amino acid, that is leucine. We have 20 types of amino acids. Again, it has given other combination, that is serine. If we keep on increasing like this, again it will give out leucine. By this, he has given a conclusion that when the combinations of nitrogen bases are different, because he kept on increasing the addition of different types of nitrogen bases, then he got different types of amino acids. Means this one individually, if you see, uracil is a pyrimidine. It is made up of a benzene ring. It is having nitrogen atoms at 1 and 3rd positions. So these can be considered as a chemical types of molecules, though they are organic molecules. Hargobind Khurana was also considered as a developer for artificial RNA or synthetic RNA. Khurana was the first person to discover synthetic RNA. Next, another scientist, Marshall Nirenbach. This person has done an experiment on cell-free system. Means he has taken some combinations of nitrogen bases in vitro conditions. Means not in the living organism, outside the living organism's body, in the test tube or in a conical flask. Has conducted experiment in cell-free system. Cell-free system in the sense in lab or in vitro conditions. Means outside the living organism's body. Here either in Nirenbach experiment or in Khurana experiment or in George Gamow proposition, we should understand that they have done some experiments and that was outside the living organism's body, and all these experiments are template free types of experiments. Means they did not use any of the pre-existing codon of DNA or RNA to give out the new types of genetic material or nucleic acids. Cell-free system for protein synthesis. That helped the code or the codon to be deciphered. PHE. Means, what is code or codon? For suppose if you take combination of nitrogen bases uracil, it is being converted. So deciphered means converting. Converted from the genetic information pattern to the protein form. Next, another person, Severo Ocavo. I pronounce this one as Severo Ocavo. You can pronounce as you like. He is a biochemist and an enzymologist. He synthesized RNA. He also synthesized enzyme that is polynucleotide phosphorylase. Polynucleotide phosphorylase. Next, this enzyme is helping or it controls the DNA or RNA synthesis. Means this enzyme is regulating the synthesis of nucleic acids. It regulates the nucleic acid synthesis. So, this total work was done in 1955 year. So, basing on all these propositions, the genetic code was framed in the or inserted into the checkerboard. So, basing on propositions of all these scientists, this enzyme polynucleotide phosphorylase was also given a special name as that enzyme is polynucleotide phosphorylase, PNPase. This is also called with this word. Now, this enzyme is also called a Severo-Ocavo enzyme, and it is a template independent enzyme. Means here for its function, it does not need any template. Template independent in function. This enzyme is also called as Severo-Ocavo enzyme. I call it as Severo-Ocavo enzyme. You can pronounce as you like. So this is about different propositions given by different scientists. Basing on the knowledge of all these, the genetic code was framed. As we have said that genetic code is nothing but a set of rules and regulations and also the correct combinations of the nitrogen bases into codons is giving out different types of amino acids. Basing on the concepts of all these persons, the genetic code was framed. It was said that it is framed in presence of Nirenbach. But the idea was including all the propositions of all types of scientists and persons who are involved in explanation of this genetic code. So if we frame out a board, checkerboard like this, as we have said that there are some rules and regulations to express or to explain this genetic code. That is first letter of the nitrogen base. So let's divide this one into 4 different chambers. the second letter. Writing of first letter means adenine in this, guanine, cytosine and uracil. But the arrangement of these nitrogen bases is also very much specific, that uracil has to be written first, next cytosine, next adenine, and next is guanine. Normally we say adenine, guanine, cytosine, uracil like this, but all these nitrogen bases should not be arranged as we like. They should be arranged only in this order. First uracil, next is cytosine, adenine, and last one should be guanine. In the same way, the second letters or second combination. Again, uracil, cytosine, adenine, guanine. But in the third letter or third type of arrangement, we should write like uracil, cytosine, adenine, guanine. Uracil, cytosine, adenine, guanine. Uracil, cytosine, adenine, guanine. Again, uracil, cytosine, adenine, and guanine. The table or the checkerboard should be written like this, and the combinations of nitrogen bases also should be expressed like this in the board. Now, first of all, if you want to fill this one, uracil and this uracil, so these are 2 in number. The first one is again uracil. So if we keep on writing like this, it will be UUC, UUA, UUG like this. And another one. So here is cytosine. Cytosine, uracil, first of all we will write cytosine. The second letter should be written, which is on the upper side. Cytosine, uracil. Third one, we should write like this, the third letter. Like C, U, C. C, U, A, C, U, G. Next, adenine, uracil, uracil. Adenine, uracil, cytosine. Adenine, uracil, adenine. Adenine, uracil, guanine. The combinations should be written like this. First of all, we should take which is on the left side, that is guanine. Second, which is on the top, that is uracil. Third one, which is on the right side, uracil. So, G like this. Let's take another one. Uracil, cytosine, uracil. Uracil, cytosine, cytosine. Uracil, cytosine, adenine. Uracil, cytosine, guanine. If we keep on filling like this, after completing of the genetic code, the combinations of nitrogen bases or the codons, it will be like this. Now here we can see total number of total codons are 64. Now, first we will see that Corona has taken some experiments, and he has done some experiments on Homo polymers and copolymers. Here Homo polymers we can take uracil, 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 guanine, guanine, and adenine, adenine, like this, and cytosine, cytosine like this. It can be any, but initially he has started his experiments on uracil. There are some rules. As we have said, genetic code is nothing but a combination of rules. So the first rule is codon is a triplet. Then only it is able to give out amino acid. For suppose one single amino acid, if they are taken for experiment, it is not giving out any amino acid. Another combination of amino acid not giving. But if another type of amino acid was taken, like adenine, 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 it is coding for lysine type of amino acid. So when 3 number of nitrogen bases are fused, then we are getting one amino acid. It means that the codons are nothing but in the form of triplets. So the first rule is codon is a triplet, and out of 64, 61 are giving out amino acids. 3 are not giving out any type of amino acid because 3 are considered as stop codons. This is one rule. Another one, see, there are many number of combinations of codons, many number of nitrogen bases, many number of triplet codons. 64 codons. Here, if we write out what type of amino acids are they giving, we are seeing only 20 types of amino acids. Means from a huge number, it is decreased to less number of amino acids. Means the information has been compressed. That conversion of huge genetic material or information, genetic information, into less number of proteins, compressing, is given a term called as degenerate, because general meaning of degenerate is also compressing from higher content to lower one, means decreasing. So basing on conversion of 64 number of codons into less number, that is 20 number only amino acids, so the codon is considered to be degenerate. First of all, we will see what are the amino acids that are coded by. We will take 4 number of equal. So all 4 codons in a chamber are coding for serine. Here, the next one is proline. The next one is threonine. The last one is alanine. Means here we have got 4 different types of amino acids. Here we will see. These are 4 here. Next, these 4 also. In this chamber, it codes for valine. Valine here. These 4 also codes for leucine. L-E-U, leucine. Next, what are again 4 different types if you see? Here it is arginine. Arginine, 4 number. Next, another number here, it is glycine. You see this one, glycine. So 4 number of codons in 4 in number, coding for one type of amino acid. What are those? Leucine, valine, serine, proline, threonine, alanine. Next here, if we take glycine and arginine, only 4 number. Next, so how many are those? 1, 2, 3, 4, 5, 6, 7, 8. 8 number of amino acids are coded by 4 same types of combinations, basing on the adenine guanine arrangement. Okay. If you see this first one, here it is coded for phenylalanine. Next, if you see in the third type of row, it codes for tyrosine. Next, here the first 3 are coding for cysteine. But if you see this combination, this one, UAA, that is opal, and UAG, amber, and UGA, amber. This is ochre, amber, and amber. These are nothing but stop codons. And this one is specially UGG. It is coding for tryptophan. Tryptophan type of amino acid. What are these coding for? In the third type of chamber, here these 4 are coding two types of amino acids. These bottom 2 are coding for acidic type of amino acids, that is aspartic amino acid and glutamic amino acid. Glutamic amino acid. Now, this is coding for lysine. This is leucine here, but this is lysine. CIL, leucine. Now, this codes for aspargine, that is basic amino acid. This also codes for another type of basic amino acid, that is glutamine. This is glutamine. And this is aspargine. Aspargine. And this codes for histidine. Histidine. So here we are seeing, I think, okay. Now, what is this coding for? This one is nothing but methionine coding sequence. That is starting codon, AUG. But the remaining 3 are nothing but coding for amino acid, that is isoleucine. Here this is also coding for leucine type of amino acid, that is leucine. So we have got here all types of amino acid, but this one is pending. These 4 are coding for arginine, but here this one AGU, these both are again coding for serine amino acid. Again, these 2 are again coding for arginine type of amino acid. So if you take, what are amino acids coded by 6 codons? By 6 codons. If you take here 2 leucine, 4 again leucine. So first answer will be leucine. Serine here 4 and here 2. So serine. Arginine here 4 and here 2. Arginine. Now the starting codon is 1. Next, acidic type of amino acids are 2. Basic type of amino acids are 2. Neutral is 1. Next, stop codons are 3. Sulfur, disulfide containing amino acids are 2. That is cysteine. Here we can see phenylalanine, tyrosine, and tryptophan. Tryptophan is coded by only one type of codon, that is UGG. All these 3 are called as aromatic amino acids. So coming to the rules. Since 64 number of codons are coding for only 20 number of amino acids, the codon or the code is, genetic code is degenerate, means it has decreased or compressed. Degenerate. I think now you understood why we use the term like degenerate. Now, the codons, when they are present in an genetic material or the nucleic acid, for suppose you take You know that DNA is like this. If the codons or an mRNA is like this, they are arranged in the form of adenine, guanine, cytosine, thymine, adenine, guanine, cytosine, thymine, like this in the sequence. Their complementary will be thymine, adenine, guanine, cytosine. On the other strand, that is different. In case of mRNA, how they are arranged? Uracil, cytosine, guanine, adenine, uracil, cytosine, guanine, etc. There is nothing comma or any type of punctuations present between the codons when they are present on the genetic material. The codons read on mRNA do not have any punctuations. And are in a contiguous, means continuous fashion. Another type of role is the codon is universal. What do you mean by this? Universal. If you take any prokaryote or eukaryote, any type of organism, these different combinations of codons are giving rise to 20 number of amino acids only. These are not giving out for any other types of amino acids. Next, the starting codon, AUG, is start codon. Anywhere you take, in either a prokaryote or eukaryote or anything, if you take, AUG codes for methionine only in eukaryotes. Start codon. Next, stop codons are 3 in any type of organism. UAA, that is ochre, UAG, amber, UGA, umber or opal. Okay, this is universally applicable type of combinations and the types of amino acids coded by different types of codons. These codons are present on mRNA. They are translated into amino acids during the translation process in presence of tRNA and rRNA, ribosomal RNA. This is about genetic code.